Hello and welcome back. So far, I've introduced the microbiota to you. I've talked about how it develops, and I've talked about its two best known effects. Now, I want to talk about how it goes wrong or how we think about it going wrong. And I want to show you some things we know that go wrong when it goes wrong. So, let's get into that. So, dysbiosis is an important word to know, and it has a two-part definition. It is the word for when things go wrong, but specifically, dysbiosis is when there is a change to the community structure of the resident microbiota. So stop. What is resident microbiota? Pause the video. Resident microbiota is the long-term residents. The community structure is the pattern of abundance of each different species or strain. Which ones are numerous? Which ones are rare? We don't know what it should be, but we know that there are some community structures that are better than others. So dysbiosis is a situation where there is a change to that community structure, a change to that balance that results in a loss of some beneficial function. So a loss of some good thing the microbiota did for us. Community structure, I like to use like a forest analogy, but I'm not going to get into too many analogies here because I don't like it when people describe the microbiota with analogies. They're good for learning, but not the point. So the example of C. diff was a great example of dysbiosis. The broad spectrum antibiotics would completely change the community structure in the colon, and C. diff would be able to colonize and grow and cause colitis. Another well-known example of dysbiosis affecting health is in bacterial vaginosis. When the vaginal microbiota community structure is completely changed, that leads to a loss of colonization resistance against chlamydia and somehow a higher susceptibility to HIV and other bad things. So that is another clear example of dysbiosis. There are a lot of other conditions that are also linked to dysbiosis that are not understood so well. So if you can look through this list, type 2 diabetes, there are various cancers, heart disease, depression and anxiety, all of these are linked to dysbiosis. It's maybe easier to imagine that inflammatory bowel disease and Crohn's disease are linked to dysbiosis, but all of these have been. And we don't know exactly what the connections are between depression and anxiety and dysbiosis. There's not some mechanism we can look at and treat with a drug. Those are what we know of in humans. In mice, we can do things with their microbiota to give them a disorder that looks like autism. And we can disrupt their microbiota in a way that makes them gain weight or lose weight. So in mice, some of their social behaviors and their body mass set point are affected by their microbiota. And in humans, that might someday be something we learn. The rest of the slides in this section are on research into the microbiota, and I've made them all curiosity slides because I just don't want to get into them too much, but I will walk you through very briefly what each one shows. So we have some experiments from mice and some experiments done in people, and you'll see the mouse experiments are real experiments where they change a variable, they affect the mice, and the mice all meet a bad end. Human experiments are more observational. We collect a person's feces and figure out what bacteria are in it, and then we try to figure out how does the person live, what do they eat, how does that connect to what's in their feces, what diseases do they have, how does that connect to what's in their feces. So cool, amazing things we found in mice that don't translate into humans or haven't yet. And then the observational things in human. This is a mouse study similar to the C. diff thing, where antibiotics are used to disrupt the gut communities. And so what this one is showing, and you'll have to take time to look at the figures if you really want to get this, but what it's showing is that they would give antibiotics to the mice and then challenge them with a pathogen to see if that pathogen could colonize. So that's vancomycin-resistant enterococcus. So they would just figure out, okay, how many of those enterococcus cells do you find in the small intestine? So in a normal healthy mouse, you find none. In a mouse that had antibiotics that you then challenged the next day, huge amounts of that pathogen would colonize. If you gave them antibiotics and let them recover for 2 weeks, you'd still find a lot of those pathogens could colonize. If you let them recover for 4 weeks after antibiotics, a huge number would colonize. And the thing is here, by 2 weeks, the mice have a microbiota. They have just as many bacteria in their colon as they did before the antibiotics, but they have different microbiota. And then this other one over here is looking at the cecum, which is more like a mouse's colon. Mice don't have a colon, but it's similar. This is an older study that looked at fecal samples from healthy volunteers up top, and it looked at which bacterial groups are in their feces. And they were hoping to see what do you expect from a healthy person? What do you expect? And what they found is that no two of these are exactly alike. But more than that, they wanted to see how does it change when someone is in intensive care. And some of these intensive care communities look very different from a healthy community. In fact, I would say all of them do. We don't know what these people were being treated for. What we know is they were admitted to the intensive care unit on day 0, and samples were taken at various different days afterwards. And if they died, they get a black dot. So on the left, these are fecal samples taken from intensive care unit patients who ended up succumbing to their illnesses. And on the right, these are fecal samples taken from intensive care unit patients who were later discharged. I used to have students look through this to try to find a pattern, and the point I could find is that there aren't any patterns, except we can tell when something is very wrong, like here. All of these are very wrong compared to the healthy ones, but we couldn't say what a healthy one should look like exactly. This study of a mouse is where they knocked out part of the mouse immune system, and what they noticed when they looked at the mouse's gut using fluorescence microscopy, they could see villi in blue with a DNA stain. So each little dot is a cell in the villi of the intestine. And then the bacteria stained green. And normally there is a 50 micrometer zone of exclusion where the bacteria really don't get through it. That's the mucus. And the bacteria that go into the mucus die. But if you remove that certain part of the immune system, the bacteria get all the way to the colon. And that's a thing to remember. We have this microbiota. We don't let them touch us. We don't want them to touch our epithelial cells. The vaginal microbiota isn't supposed to touch the vaginal epithelium, but in bacterial vaginosis, it does. And then this one is complicated. This shows each stack here. If you'll notice over here on the left, that is a stack of black and yellow bars. And each of those bars represents a type of bacteria and how abundant it is. And if it is bright yellow-green, it's abundant, and if it's black, it's rare. And then notice, if you look very carefully at A, I see 1, 2, 3, 4, 5, 6, 7, 8 different columns of bars. Well, that's 8 different samples from one patient's colon microbiota. And there are 5 from patient B and 5 from patient C. And these blue bars represent a time where these patients received ciprofloxacin, so Cipro, that antibiotic. Notice how the microbiota appeared to change during Cipro treatment. And then notice, did they seem to go back to normal? This would look very different with other antibiotics, and I don't have a way to tell you whether these microbiota regained all their functions, but I can tell you they certainly look like they came back to where they were. In the next video, I'll talk about the vaginal microbiota and bacterial vaginosis, and that is also a complicated topic that I will have to pick up in the next video. So thanks for your attention. I will see you later.